is Carmen Collado. I'm a researcher staff at the Institute of Agrochemistry and Food Technology belonging to the Spanish National Research Council. I would like to thank the opportunity to explain my work to the Biostime Institute Nutrition and Care. And today we are going to talk about the role of the maternal microbes on infant health. It has been recognized that maternal microbiota is one of the most essential factors determining maternal child health outcomes. Early life exposures before pregnancy, during pregnancy, and during lactation are going to have an influence to the infant in terms of the microbial development, immune system maturation and development, and also the metabolism. We cannot define the healthy microbiota or infant healthy microbiota. However, thanks to the epidemiological data, we can consider an adequate infant microbiota from those babies born by healthy moms, vaginal delivery, at full term, and exclusive breastfed during at least 6 months of life, according to the World Health Organization recommendations. Maternal microbiota can be exposed to the infant or to the fetal development in 3 key time points: during the pregnancy, at birth, and during the lactation. And we are going to explain a little bit these critical steps where humans are exposed to maternal microbes. The first contact during the pregnancy. Well, we know that there is a huge debate about if humans are exposed to the bacteria before birth, and there is a contradictory evidence available right now in the literature. Then the question is, does the fetus reside in a sterile environment? Well, some studies are showing that placenta microbiota harbors bacteria. Others also show that bacteria or DNA from bacteria are present in amniotic fluid and cord blood. Fetal membranes and colostrum contains as well bacteria, and different studies in literature also highlight the presence of specific microbes in meconium. Furthermore, microbial communities have been identified in the oral cavity and nasopharynx intern newborns. However, other studies, including this recent one published in Nature Microbiology, suggest that fetal microbiota doesn't exist. The infants are exposed to the bacteria just after birth, when they are exposed to the maternal microbiota and environmental microbiota. In this study, they use a really nice and fashion protocol where they collect the fecal sample with a swab in a specific C-section deliveries, as you can see in the picture. They found mainly skin contaminants, mainly Staphylococcus epidermidis, and they don't detect bacteria in most of the samples. On the other side, different studies highlighted that the placenta microbiota is not present, and the DNA from bacteria that is detected in the different studies belongs to the DNA contaminants in the reagent kits are also in the environment. Then still the question is open and the debate is open. But the fact is that maternal microbiota is changing over the pregnancy. Shifts on the maternal microbiota has been implicated in the metabolic and immunological adaptations during the pregnancy. Different studies have been highlighted that the oral microbiota in the pregnant woman changed with an increase in proteobacteria or pro-inflammatory bacteria, Polyphomonas gingivalis and Agrigibacter. Also, that microbiota changed over the pregnancy with an increase in actinobacteria, mainly bifidobacterium, from the first trimester to the third trimester of the pregnancy, as well to an increase in proteobacteria, pro-inflammatory bacteria in parallel to the weight gain according to the fetal development. Vaginal microbiota also has been reported to be changed during the pregnancy with an increase in lactobacillus and a decrease in alpha and beta diversity. Then moms are changing the microbiome during the pregnancy with potential implications for the fetal development, but still we need more data and more evidence. In our team, we are quite interested to understand which would be the key role of the diet in those changes in the microbiota during the pregnancy. We ran an observational and cross-sectional study at birth where we compared the microbiota of the woman according to the Jensen channel distance and PAM method, we cluster or we separate the microbiota of the moms, and interestingly, we found 2 specific clusters, each of one associated with a specific bacteria. And then trying to identify if these microbiota were linked with diet, also we found that clusters had different dietary profile. For example, cluster 1 is characterized as a higher diversity and richness and higher abundance of Prevotella, Finegolgi, and other bacteria, and as well characterized by a higher intake of specific fatty acids. However, the cluster 2 was associated with a lower diversity and richness, and also with a higher presence of Blautia, Roseburia, Faecalibacterium, many of them known as a short-chain fatty acids producers, and also bacteroides. This cluster 2 was also associated with higher intake of fiber, omega-3 fatty acids, and some polyphenols. The interesting thing is that these microbial and dietary clusters in mothers were linked to the infant microbial development, and the impact of these clusters were even observed at infant growth up to 18 months of life. We also identify that moms with higher intake of fat had higher sonulin in the feces, in the intestine. Sonulin has been considered as a marker for intestinal permeability. Then higher lipid consumption was associated with higher permeability and higher sonulin presence. But these changes in mothers were also reflected on the neonate, and we observed that moms with a higher intestinal permeability had a neonates with a characteristic microbial composition, mainly adult-like type bacteria in the neonates. We know that infant gut microbial colonization is a stepwise process where some species are providing an adequate environment for the next ones. Then to have or to harbor adult-type bacteria in the naive or immature intestine has been associated with higher risk of a specific disease. Then further studies are needed to understand what are the impact or the biological significance of these adult-type bacteria in the gut, in the neonatal gut. The second contact of humans with maternal microbiota is the birth. We know that most of delivery, C-section, and vaginal are playing an important role in terms of microbial exposition, but also gestational age, maternal health, hospitalization, exposition to antibiotics are going to play an important role to the infant microbiota. It's at birth when the humans are exposed to a massive amount of bacteria, mainly from the vaginal tract and gut microbiota from the mothers. And this happened mainly in infants born vaginally. That's the reason that vaginal delivered babies harbor a microbiota more similar to mothers compared to the C-section babies who harbor a microbiota more associated or more similar to oral bacteria, environmental bacteria, and skin bacteria, and also with a delay in bacteroidetes and lower presence of bifidobacterium. Then it is needed to understand that mode of birth, it's disrupting the maternal transference of microbes to the neonate, and in the case of the C-section, is even more because of the exposition of antibiotics. In our team, also, we wanted to understand or to know more about the environmental factors. Then we compare babies born in a hospital, vaginally, but also under C-section, compared to infants born at home. And we identified different microbial evolution during the first month of life, where vaginal and home birth vaginal babies harbor higher levels of bifidobacterium and bacteroides compared to the C-section. That shows, in agreement with literature, a delay in bacteroides and an increase in potential proteobacteria. But not just the gut microbiota is influenced by mode of birth and other factors. We also study the impact of the mode of birth in the oral cavity of the neonates. We observe a significant different trajectories up to 6 months of life in infants born vaginally compared to the C-section. After 6 months, we didn't observe the effect of mode of birth in the oral cavity, and this may explain because these babies are exposed to the complementary food, or maybe they are tending to the kindergarten. The same happened to the breastfeeding habits. We observe a distinct trajectory in the infants partially breastfed from birth to 7 years of life compared to infants non-breastfed. And this allows me to start with the next exposition, which is the lactation. It's clear that diet shapes the gut microbial community, and for example, exclusive breastfed infants harbor higher bifidobacterium levels in their gut, and also the cessation of the breastfeeding practices rather than introduction of the complementary food had a really big impact on the microbial composition and diversity. We also studied the influence of the donor milk in preterm babies. We identified that babies fed with On's mother milk and donor milk had a similar microbial profile in their gut compared to the formula-fed infants. The donor milk and maternal mother's milk gut microbiota were similar, but still different. And the main reasons would be explained by two main points. The first one is that donor milk suffered a heat treatment, pasteurization, to assure the quality and the safety for the preterm babies. And we know that heat treatments may disrupt and decrease specific bioactive compounds present in human milk. Furthermore, also we know that most of the donor milk are coming from term deliveries, and we know, and literature is available of the different data, that milk from term deliveries are totally different in macronutrients, micronutrients, and also bioactive compounds than those observed in preterm or in premature birth. Then we found these differences, but still donor milk had a similar microbial profile in the preterm babies. But what is in human milk? Why the effects are so important? Then we know that human milk is the gold standard for infant nutrition, and it's more than just nutrition because it contains as well different bioactive compounds. There is a really complex to work with human milk because human milk changes during the lactation stage. The composition is totally different from colostrum to transitional milk to mature milk. And also we know that human milk changes within 1 day, within breast, and also within feds. Then this is so difficult to study the human milk. There is a huge information about the different potential bioactive compounds that are present in human milk, and thanks to the development of the techniques, we know more about these compounds, as for example, protein, peptides, nucleotides, hormones, growth factor, immunomodulatory compounds, anti-inflammatory agents, hormones, oligosaccharides, microbes, and also other compounds. And all these bioactive compounds are really in few quantity or absent in infant foods. These specific bioactive compounds have specific targets in the neonatal intestine and intestinal epithelia, at the enteric nervous system, and also at the mucosal immune system. We are going to talk about the microbiota because the topic is related to maternal infant microbial transfer, and we know that human milk is a constant source of microbes. Thanks to the culture-dependent and independent methods, we know that there are different species present in human milk, and thanks to the new technologies, mainly the sequencing, we know that there are more than 800 different species present in human milk. Mainly, most of them are belonging to Staphylococcus and Streptococcus genera, but also we can identify some lactic acid bacteria, Bifido, but also proteobacteria as Escherichia coli, Klebsiella, among others. The quantity of total bacteria present in human milk is not so big, it's quite small. It's around 10 times up 4 and 10 times up 5 colony-forming units per milliliter. But if we think that baby is feeding around 800 milliliters or 1 liter per day, there is a constant source of these microbes that are supporting the initial pioneers that we receive at birth from our mothers. What about the potential functions that these bacteria are exerting in the neonatal gut? Well, we can identify or we can visualize the same functions that in the gut microbiota. These microbes are exerting properties at the structural level because they are promoting the intestinal homeostasis, they are promoting the adequate maturation of the intestinal gut. At the metabolic level, because some of them are able to produce short-chain fatty acids, but also some vitamins needed for infant growth. And this bacteria also exerts a protective effect because they are interacting with the immune system, but also they are avoiding that some potential pathogens or toxins from the environment and from the diet may infect the cells and produce some disease. But the presence of bacteria in milk is not a single hallmark in humans. There are different reports showing that bacteria also is present in milks from other mammals. Interestingly, there is a huge variability, inter and intra variability in milk microbiota. I always love to show this work from Shelley McGuire team because it's really clearly identified. Well, you can see different profiles. Each column represents one lactation time, colostrum, first column, second column transitional milk, and third column is mature milk, and different colors represent different bacteria. Then you can see how the profile from mom S7 is totally different from the mom S8. However, despite this variability, when we compare the milk microbiota with other human niches, we can see how milk microbiota clustered in a totally separate cluster compared to the oral microbiota, vaginal microbiota, and meconium microbiota, suggesting that there is a milk microbiota. Which are the factors that are affecting the human milk microbiota? Well, the same ones that we mentioned at the beginning, the same ones that may influence the maternal microbiota. And among them, maternal diet, maternal health, antibiotic exposure or other medicine consumption, gestational age, lactational time, mode of birth, mainly associated to the antibiotic exposition, and environment. Then there are many factors that can also modulate the maternal microbiota and reflected in milk microbiota. Among them, I would like to highlight the influence of the maternal diet on milk microbiota. This is a work that was run in my team, and we also identify these two different clusters according to the diet that are shaping the milk microbiota. We observed that Staphylococcus and bifidobacterium were associated with higher intake of carbohydrates, while Streptococcus present was associated with higher intakes of N3 profile. Interestingly, we found that according to the maternal diet profile and maternal microbiota, the influence of the antibiotics were different. Well, in brief, if you are eating well, the impact of the antibiotics with the C-section would be less intense than if you are eating bad. And this opened new possibilities to develop dietary strategies in future. Of course, we need to think that there are other bioactive compounds that can interact directly with the microbes, for example, human milk oligosaccharides, but also other factors as growth factors, cytokines, and others, protein peptides, and fatty acids. And still, little evidence is known, or little information is known about these complex relationships among all of them. Another thing that I would like to point out in this presentation is that most of the science is focused on the bacteria, but what about other organisms? In our team, do a casual finding, we identify the presence of fungi and yeast in human milk samples from healthy mothers. We use different methodologies, classical culture approaches, fluorescent hybridization under the microscope, and also sequencing, and we identify a really wide spectrum of different fungi and yeast. Interestingly, we identified the presence of Saccharomyces cerevisiae, which is a well-known yeast used in bread, for example. Then maybe food or diet would be one of the most important sources to shape the fungi and yeast presence in human milk, but also in infant gut. Another study, recent study run in really big cohort in Canada led by Megan and her team, they also identified the presence of fungi in healthy breast milk. They identified Candida alternaria and Rhodotorula. The interesting thing of this study is that they associate the presence of the fungi with a multiple outdoor environmental features, as for example, city, population density, and season, and also link it with a maternal health status, as for example, maternal atopy, and also to the impact of the antibiotic, because antibiotic removes bacteria, but this would provide an advantage for other organisms as yeast that they are not affected by these antibiotics. Then a lot of things are now suggesting that diet and environment are shaping our microbes. Then factors that can modulate the microbiota, or in another way, alterations in maternal microbiota, are going to be transferred to the neonate with an impact to infant gut maturation and infant immune system response, with an effects in short and long-term effects on infant health and adult health. Then the question is, if we know that the alterations are bad, can we modulate the maternal microbiota to try to minimize this altered transference in terms of the microbiota? And if yes, when and where and how? Do we need to do it during pregnancy or even before, or just during lactation to modulate the milk microbiota? Or maybe we need to focus in both, in moms and in neonates? And if yes, can we use dietary counseling or specific foods or specific food supplements, as for example, probiotics, prebiotics, or symbiotics? Still, we have more questions than answers, and we need more information on these things. But just to show some interesting things, we know that probiotic intervention early in life are able to reduce the risk of atopic disease prevalence in children population at high risk. We know that probiotics during pregnancy are able to modulate the number of bifidobacterium in the neonate, but also to control and to modulate the glucose metabolism during the pregnancy and reduce the prevalence of gestational diabetes. And also, different studies show that probiotics during the gestation are able to modulate the immune gene expression in the placenta and in the fetal gut. We also know that probiotics are able to modulate specific compounds present in breast milk, as for example, TGF-beta2, that is an anti-inflammatory cytokine that promotes the oral tolerance and promotes the IgA production, that helps to reduce or to allow to reduce the risk of atopic eczema and allergy problems. But also, a recent study also highlighted that probiotics are able to modulate the oligosaccharide composition and profile in breast milk. And not just in moms, there are information that probiotic intervention and also some dietary counseling are able to improve the sperm quality. And this is something that would help us to improve, for example, the reproductive treatments. Then again, there are more questions than answers, and this is a really hot topic because human health starts in utero. To conclude, there is an interplay between diet, environment, and microbiota during the pregnancy, and this perinatal period is critical because it has relevant consequences for infant and adult health. Then it is needed to increase our knowledge on the role of the maternal bacteria for infant microbiota, because this knowledge will help us to develop new diet strategies or maybe other type of strategies based on microbial modulation aimed at the beneficial microbiological, immunological, and metabolic programming of child health. Thank you to my team. Thank you for the funding that we get to run these studies. And of course, thank you again to Bing for this invitation. Thank you very much to all of you.